Hello guys, welcome to my channel. So as you know, in the previous video we studied about the polysaccharide, that is starch and glycogen, correct? Right, so moving further in the polysaccharide, so we are studying about the homopolysaccharide. So the next is the cellulose, right? So I'm not doing this. I'll just remove this and put here right now. The next is cellulose for today. So what is cellulose? Cellulose is a tough, fibrous, water-insoluble substance is found in the cell walls of plants. So this is found in the cell wall of plants, particularly in the stalk, stems, trunks, and all the woody portions of the plant body. So the cellulose constitutes much of the mass of the wood, and cotton is almost pure cellulose, right? So the mass of the cellulose, where it's in the mass of the wood, and where it's cotton, it's almost pure cellulose. Like amylose that we studied here, amylose, right? The cellulose molecule is linear, the same linear and unbranched, so it is not branched like this one. It's unbranched homopolysaccharide that is consisting of 10,000 to 15,000 D-glucose units. But the very most important difference is the cellulose glucose residues have beta configuration. Like this has alpha configuration, but the cellulose has a beta. So this has beta configuration. This has beta configuration, right? Whereas in amylose, right, that we see here, that has alpha configuration. So the glucose residues in cellulose are linked. This is alpha 1, so these are linked as beta 1 to 4. So these are like beta 1 to 4 by glycosidic bonds, in contrast to this alpha 1 to 4, right? This difference causes individual molecules of cellulose and amylose to fold differently in space, right? What this difference makes? Cellulose and amylose fold differently in the space, giving them very different microscopic structure and physical properties as well. So the tough, fibrous nature of cellulose makes it useful in such commercial products such as cardboards and insulation materials, and it is a major constituent for cotton and linen fabrics, right? And cellulose is also starting material for the commercial production of cellophane, rayon, or lyocell, right? So that was cellulose. So the glycogen and starch ingested in the diet are hydrolyzed by alpha amylases, right? So the glycogen and starch that we ingest in our diet are hydrolyzed by alpha amylases and glycosidases. Okay, so not removing everything. Maybe it might be useful. So glycogen and starch ingested in our diet are hydrolyzed by alpha amylases and glycosidases. Amylases and glycosidases, right? And these are the enzymes in saliva and the small intestine. So these are enzymes in saliva and small intestine, right? So what they do, they break the alpha 1 to 4 glycosidic bond between the glucose unit, right? That's the function of this alpha amylases and glycosidases. Their enzymes are present in saliva and small intestine. They break this glycosidic bond between alpha 1 and 4, right, between the glucose units. So most vertebrate animals cannot use cellulose as a fuel source. Why? Because the cellulose, I wrote that, but the cellulose as beta 1 to 4, right? We just studied now linkages. So that's why if we use cellulose, these enzymes cannot break the cellulose into separate units, right? So termites readily digest cellulose, but termites, right? They can readily digest cellulose and therefore wood that they are eating, but only because their intestinal tract harbors a symbiotic microorganism, and that's why they can digest cellulose. Which microorganism? That so the microorganism Trichonympha. Right, that is Trichonympha. So this is the microorganism that secretes cellulase, which hydrolyzes the linkage beta 1 to 4. Beta 1 to 4 that hydrolyzes the beta 1 to 4 linkages. That's why the termites can digest or hydrolyze the cellulose. So the molecular genetic studies have revealed that genes encoding cellulose degrading enzymes are present in genomes of a wide range of intervertebrate animals that are including arthropods and nematodes. So there is one important exception to the absence of cellulase in the vertebrates: ruminant animals. Why it's called ruminant animals, such as like cattle, sheep, goats. So these animals harbor a symbiotic microorganism in the rumen. So what is rumen? In the rumen, rumen. So the rumen is the first of their four stomach compartments. So there are four stomach compartments in this kind of animals, and the first compartment is the rumen, and they harbor the symbiotic microorganism that can help hydrolyze the cellulose, so allowing that animal to degrade the dietary cellulose from soft grasses but not from the woody plants. And fermentation in this rumen, in the part of the first compartment of stomach, yields acetate, propionate, and b-hydroxybutyrate, which the animal uses to synthesize sugars in the milk like lactose. Right, so the biomass that is rich in cellulose can be used as a starting material for fermentation of carbohydrates to ethanol. That means that is to be used as a gasoline additive. That is like a switchgrass is a common biofuel crop. Sorry. So the annual production of biomass on Earth that is accomplished by primarily by photosynthetic organism is the energetic equivalent of nearly 1 trillion barrels of crude oil. So when converted to ethanol by fermentation, right? Because of their potential use in biomass conversion to bioenergy, cellulose degrading enzymes such as cellulase are under vigorous investigation. So like that's why the scientists are investigating the cellulase, so that can help the biomass conversion to bioenergy. That is very useful. So the supramolecular complexes called cellulosomes found on the outside surface of bacterium Clostridium cellulolyticum include the catalytic subunit of cellulase, along with the proteins that hold one or more cellulose molecules to the bacterial surface, and a subunit that binds cellulose and positions in the catalytic site. Alright, so that was cellulose. And the next one is chitin. So I will write it down here. Chitin. Right, so this is a linear homopolysaccharide. This is just a linear, we'll write it down, linear homopolysaccharide. This is a linear homopolysaccharide, but this is composed of an acetylglucosamine. Right, composed of an acetylglucosamine. Right, this is composed of an acetylglucosamine residues. This is composed of alpha glucose, but this is composed of an acetylglucosamine residues, and the linkage is beta 1 to 4, the same as that we studied in cellulose. So the only chemical difference from the cellulose is the replacement of hydroxyl group HC2 with an acetylated amino group. Why? Because it is acetyl glucosamine. So that's only the difference between the cellulose and the chitin, that the hydroxyl group at C2, right? So this is 1, because this is 2. So the hydroxyl group that is here at C2 is actually replaced by an amino group. That's why it's called an acetyl glucosamine. Right? The chitin forms extended fibers similar to those of cellulose, and like cellulose cannot be digested by vertebrates. Right? So this chitin is also, what it forms is it forms extended fibers like those of cellulose, and same like cellulose, the vertebrate animals cannot digest this cellulose or chitin as well. So the chitin is the principal component of hard exoskeletons of nearly 1 million species of arthropods, that is like insects, lobsters, crabs, right? These are the example of chitin. So the where chitin is present is the principal component of hard exoskeletons of nearly 1 million species of an arthropods, right? And is probably the second most abundant polysaccharide next to cellulose in nature. So the first abundant polysaccharide is cellulose, but the second one is this chitin. That's why we are studying it. And this is estimated 1 billion tons of chitin are produced in biosphere each year. Right, on your screen you can see the picture. That's the formula of chitin, and that's the beetle, right? And the surface, outer surface, you can see that's the exoskeleton of that is made up of chitin. Alright guys, so that was cellulose and chitin. Last, in the previous video we studied about starch, glycogen, right? And dextrans as well, that I removed from here. And in this topic we just studied about the cellulose and the chitin. So in the next video we'll study about the steric factors and hydrogen bonding that influence homopolysaccharide foldings. So we'll study about the foldings. Alright guys, thank you guys for watching, and don't forget to subscribe the channel. Thank you.